Hello, and welcome to my video series on algorithms. This is part 4 of Module 6, Dynamic Programming. In this video, we will discuss the sequence alignment problem. The sequence alignment problem involves comparing two sequences to measure their similarity, which has crucial applications in many fields like computational biology. One important use is to determine evolutionary distance between species. For instance, comparing the morphological features of similar animals like primates can be challenging, since they often share traits like fur, tails, and the ability to climb trees. Instead, we can compare their genomic sequences and use their similarity to quantify evolutionary distance. This approach allows us to construct evolutionary trees that can illustrate the relationship among the primate species. Another key application of sequence alignment is in inferring protein function, where similar protein sequences often indicate similar functions. For example, in Dr. Russell Doolittle's landmark paper, he identified a significant sequence similarity between a protein in a cancer-causing virus and the human growth factor. The discovery linked tumor formation to the abnormal growth of cancer cells, laying the foundation for much of today's cancer research and treatment strategies. Now, let's formally define the sequence alignment problem. We are given two sequences, S1 and S2, both derived from the same alphabet. Here, we assume the DNA alphabet of A, C, G, and T, though the problem can be defined with any alphabet. Our goal is to quantify the similarity between S1 and S2 and visualize the differences. Note that these two tasks are essentially the same. This is because quantifying the similarity involves identifying the differences, and visualize them is just a way to represent this information. Therefore, we will not discuss this task separately. In this video, we will cover necessary technical details to address both aspects. Let's begin with defining the sequence similarity we want to measure. Consider two sequences, TGC-ATAT and TCAG-ATGT. The easiest way to measure their similarity is using the Hamming distance, which compares the two sequences letter by letter and count the number of mismatched letters. In this case, the Hamming distance between the two sequences is 7, where we marked the mismatched letters red. However, a key limitation of the Hamming distance is that it can overestimate the difference if the sequences are not properly aligned. For instance, by aligning the sequences better, as shown in the right-hand side of the arrow, the number of mismatched letters is reduced to 4. The adjusted sequence similarity measurement is called edit distance, which counts the number of edits required to convert one sequence into the other. The edits are implied by the different letter pairs that are marked red. In our example, the first edit would be deleting G, followed by converting T into G, inserting G, and inserting T. Notice that properly aligning the letters plays a critical role in computing the edit distance. By aligning the letters, we mean inserting the dashes, or gaps, into the original sequences to match identical letters in the same columns as much as possible. Thus, computing the edit distance can be broken down into two steps. First, aligning the letters, then computing the Hamming distance. The representation shown on the right-hand side of the arrow, which highlights identical and mismatched letters, is called the alignment between two sequences. Hence, we name this problem sequence alignment. Let's also clarify two important points regarding the edit distance. First, the edit distance focuses on finding the minimum number of edits. Searching the maximum number of edits is meaningless, as one could keep changing the same letter back and forth. Another reason for finding the minimum is because we assume that the nature can always find the most efficient way to evolve the DNA. Second, there are three specific types of edits allowed in this context. The first type of edit is called mutation, which changes a letter in S1 to another letter in S2. The second edit is called insertion, which inserts a letter in S1. The third edit is called deletion, which deletes a letter in S1. It is important to know that insertion and deletion are symmetric. An insertion in S1 can be viewed as a deletion in S2, and vice versa. By convention, we take S1 as the reference and name the edits accordingly. Here, we demonstrate an example of mutation. It changes the letter T in S1 into G in S2. This is an example of insertion. From the alignment, we can see that a gap in S1 is aligned to the letter G in S2. This indicates that there was originally no letter in S1 at that position. As a result, to convert S1 to S2, we need to insert the letter G to S1. This is why this edit is called insertion. Similarly, here we have an example for the deletion edit. Let's first explore whether the sequence alignment problem can be solved using a greedy algorithm. We propose three greedy strategies, each corresponding to one of the three permitted edit operations. We could take a mutation-first strategy, meaning that we preferentially apply the mutation edit when seeing two different letters. Similarly, we also propose the insertion-first and deletion-first strategies. Using the mutation-first strategy, the two sequences shown here have a distance of 7. Notably, this distance is identical to the Hamming distance. This happens because the mutation-first strategy tends to avoid gaps, thus becoming identical to the calculation of Hamming distance. As a result, this strategy does not correctly compute the minimum edit distance. Next, let's try the deletion-first strategy, which deletes a letter in S1 whenever the two letters being compared are different. The first mismatch occurs at the second column, where S1 has a G and S2 has a C. Following the deletion-first strategy, the algorithm deletes the G in S1, leading to the alignment shown in the middle. The algorithm continues until it encounters the second mismatched pair, T in S1 and G in S2. It then aggressively applies deletions in S1, attempting to match the G in S2 with another G in S1. However, it exhausts all letters in S1 without finding another G. At this point, the algorithm must insert the remaining letters from S2, resulting in the alignment shown on the right. This results in a distance of 9, with too many unnecessary gaps, making the solution suboptimal. Due to the symmetry of the problem, we can infer that the insertion-first strategy would also fail to provide an optimal solution. In conclusion, no simple greedy strategy can correctly solve the sequence alignment problem, and we will turn to dynamic programming for a better solution. To solve the sequence alignment problem using dynamic programming, we first need to define subproblems. Given the two sequences, we would naturally begin with examining the first letter in S1 and the first letter in S2. If they are the same, we move on to the next pair of letters. If they are different, we must decide which type of edit operation to apply: mutation, insertion, or deletion. As we continue through the sequences, we will be comparing the ith letter in S1 and the jth letter in S2, deciding which edit to take. Note that I and j may not be equal, as we may take different numbers of insertions and deletions, and consume different numbers of letters from the two sequences. For simplicity, we denote the ith letter in S1 as S1I and the jth letter in S2 as S2J. In this case, the decision we make depends on the parameters I and j, which will help defining our subproblems. Specifically, define Eij as the edit distance between the I-long prefix of S1 and the J-long prefix of S2. Let's visualize this reasoning process using a simple example. We represent the two inputs as bars. The yellow sections indicate aligned letters, while the pink sections represent letters that still need to be aligned. At the start, we have aligned the first I-1 letters in S1 and the first J-1 letters in S2. By definition, the edit distance between these prefixes is E-I-1, J-1. Now, we are comparing the letters S1I and S2J, highlighted by black boxes. Before making any decisions, these letters remain unaligned. If S1I and S2J are the same, we can align them directly and move on to the next pair. If they are different, we can take one of the three permitted edit operations. In the first case, we can apply a mutation to convert S1I to S2J. This will align S1I to S2J and color them yellow, as they are now aligned. Because both S1I and S2J are consumed, the resulting edit distance is Eij. Second, we could also apply an insertion to align a gap in S1 to S2J. In this way, S1I will remain unaligned, which stays pink, and S2J will become aligned, which will become yellow. As the edit only consumes S2J but not S1I, the resulting edit distance is E-I-1J. Symmetrically, we could also apply a deletion and obtain the result E-I-J-1. With this intuition in mind, we can move on to verify the overlapping subproblem and the optimal substructure properties. Verifying these properties will confirm if dynamic programming is suitable to solving the problem. First, let's verify the overlapping subproblem property. From the figure, we observe that the solution of E-I-1, J-1 is reused in constructing the solutions for Eij, E-I-J-1, and E-I-1J. As a result, E-I-1, J-1 is referenced by multiple subproblems, making it an overlapping subproblem. Next, let's verify the optimal substructure property. Consider the case of applying a mutation to align S1I and S2J, which takes exactly one edit. Thus, we can express the relation as Eij equals E-I-1, J-1 plus 1. Similarly, if we opt for insertion, the equation becomes E-I-1J equals e(i-1) j-1 plus 1. And for deletion, we have ei j-1 equals ei-1 j-1 plus 1. In all of these equations, the left-hand side is maximized if ei-1 j-1 on the right-hand side is maximized. Therefore, this problem does exhibit the optimal substructure property. After verifying that the problem can be solved using dynamic programming, we can move on to design the specific algorithm. We will begin by establishing the partial ordering of the subproblems. Then, we will write the top-down recursive function and perform the bottom-up tabulation. The partial ordering of the subproblem can be directly obtained from our previous analysis. Specifically, we have derived 3 equations that describe the relationship among the subproblems. They are eij equals ei-1 j-1 plus 1, ei-1j equals ei-1 j-1 plus 1, and ei j-1 equals ei-1 j-1 plus 1. Thus, for 2 subproblems, eij and emn, we say eij depends on emn if any one of the following conditions holds. We could have I equals m-1 and j equals n, or I equals m and j equals n-1, or that I equals m-1 and j equals n-1. We can now combine the 3 established equations to formulate the top-down recursion. We need to adjust the indexes used in these equations to make the left-hand side term become eij. Additionally, we also need to consider the case where S1i is the same as S2j. To handle this, we introduce a variable delta to adjust the added cost depending on whether S1i is the same as S2j. Specifically, if S1i and S2j are the same, delta will be 0, meaning no edit is required. On the other hand, if S1i and S2j are different, delta will be 1, indicating that a mutation is needed. Finally, we can implement the bottom-up tabulation approach. Since the subproblems are defined by 2 parameters I and j, we need to allocate a 2-dimensional table to store the results. Specifically, we will allocate a 2D matrix M, where the entry mij will store the value of eij. Given the 2 input sequences, TGC ATAT and TCAG ATGT used at the beginning, we will allocate a 7x8 matrix, where 7 and 8 represent the length of the input sequences. Based on the recursive function, we know that the entry eij depends on 3 other entries. The left entry eij-1, the top entry ei-1j, and the top left entry ei-1 j-1. For this dependency structure, it is important to properly handle the boundary cases. For example, when computing E11, all its dependencies fall out of bound. To properly handle the boundary cases, let's visualize here the specific scenarios they represent. Specifically, there are 3 alignments that can lead to the solution of E11. In the first alignment shown on the left, 2 gaps at the beginning of the sequences are aligned. Here, we consume 1 letter from each sequence to construct the solution for E11. In the second alignment shown in the middle, we can introduce a gap at the beginning of the first sequence to construct the solution for E11. By symmetry, for the third scenario, we can introduce a gap at the beginning of the second sequence to construct a solution for E11. We observe that all three alignments involve the use of gaps at the beginning of the sequences. According to definition of E, these alignments correspond to E00, E10, and E01, respectively. In this case, we need to allow both parameters, I and j, to take the value of 0. This requires us to include an additional row and column in the DP table to store the results for when the parameters are 0. The extra row and column, highlighted in green, represent initial gaps in the sequences. With this adjustment, the dimension of the DP table will become L1 plus 1 by L2 plus 1, where L1 and L2 are the length of the input sequences. Once the values in the green row and column are computed, we can calculate the remaining entries without the out-of-bound issues. To compute the value of E00, we know that it corresponds to the case where two beginning gaps are aligned. Since gaps are identical and no edit is required, the value of E00 should be 0. To compute the value of the first row, note that it corresponds to the case where beginning gaps in S1 are matched with the prefix of S2. For example, E04 represents the alignment where the first four letters of S2 are matched with gaps in S1. In this scenario, 4 edits are needed, meaning that E04 equals 4. More generally, for all I, we have E0I equals I, meaning that the number of edits to match the I long prefix in S2 with gaps in S1 is I. By symmetry, the first column represents cases where a prefix of S1 is matched with gaps in S2. Following the same logic, we have EI0 equals I for all I, meaning that the number of edits to match the I long prefix in S1 with gaps in S2 is I. Now, we can fill in the values for the first row and column, as shown in this figure. With the boundary values in place, we can compute all remaining entries in the table row by row, using the established recursive function. We summarize the algorithm as pseudocode here. Lines 2 to 4 handle the boundary cases, computing the values for the first row and column of the DP table. Lines 5 to 11 compute the remaining entries based on the recursive function. Specifically, line 7 compares the two letters S1I and S2J and determines the value of delta. Line 8 evaluates the three permitted edit operations and records the minimum edit distance. And line 9 stores the decision in the traceback table T. Line 12 calls the traceback function, which uses T to reconstruct the optimal alignment corresponding to the minimum edit distance. Here, we present the pseudocode for the traceback function. Lines 5, 7, and 9 handle the mutation, insertion, and deletion cases, respectively. Note that in line 12, we reverse the alignment of the two sequences. This step is necessary because the traceback is performed in reverse order, where the alignment of the suffixes was constructed first. Hence, by reversing the result, we can obtain the alignment in the original direction. Finally, let's run the algorithm on a given example to verify if it produces the desirable alignment. First, to compute E11, we need to consider three cases. We know that S11 and S21 are both T, meaning that E11 can be obtained from E00 without any edit. In this case, E11 can be the same as E00, which is 0. E11 can also be obtained from E10 by applying an insertion. In this case, E11 will be E10 plus 1, whose value evaluates to 2. Similarly, E11 can also be obtained from E01 with a deletion, which will make E11 2. Next, we select the minimum among these three values, making the final result for E11 0. Additionally, we record in the traceback table T that the optimal value of E11 was derived from E00, as indicated by the red arrow. For the second entry E12, if we compute it from the top left entry E01, the result will be 2. This is because S11 is different from S22, requiring a mutation to convert one to the other, which sets delta to 1. Therefore, E12 equals E01 plus 1, resulting in 2. However, the minimum value of E12 can be achieved from the left entry E11 by adding an insertion operation. In this case, the final value for E12 is 1. Occasionally, taking different operations may result in ties. For example, computing E16 from the top left entry E05 yields a value of 5. Meanwhile, computing E16 from the left entry E14 also results in a value of 5. In such cases, we can break the ties arbitrarily, for example, by choosing the top left entry. Throughout the rest of the table, when ties occur, we only record one of the possible decisions. Following the same procedure, we will complete the entire matrix, as shown in the figure. Afterward, we will perform the traceback to recover the optimal edit operations that result in the minimum edit distance. The recovered optimal edit path is highlighted here. With the set of optimal edit operations, we can now reconstruct the alignment. Since the traceback is performed backwards, the first edit operation we encounter is the arrow from E78 to E67, indicating that one letter from each sequence is consumed. As a result, we align T and T, the last letters of both sequences. For the same reason, we align A and G, the second last letters of both sequences. The second last edit operation is an insertion, which consumes a letter in S2 but without consuming any one from S1. Therefore, we align the letter C from S2 to a gap in S1. As you can see, the alignment is reconstructed in the reverse order alongside the traceback. This is why the reconstructed alignment needs to be reversed before being output. The time and space complexities of the algorithm are both O L1 times L2, where L1 and L2 are the length of the two input sequences. This is because the major computational effort is spent on filling the DP table M. Calculating each entry in M requires evaluating 3 cases, where all can be performed in constant time. As a result, the time and space complexities both become O L1 times L2. Thank you for watching and see you next time.